Welcome to today's session of the plant physiology and metabolism. In the previous lecture, we have just given a brief introduction of the amino acids, their classification, their role and functions. So now today we will discuss about the biosynthesis of the all 20 amino acids. The majority of amino acids are produced through transamination reaction. In transamination, the amino group or NH2 is transferred from an amino donor compound to the carbonyl position of an amino acceptor compound. Commonly, amino acids like glutamine or glutamic acids serve as the donor compound, while a 2-keto acid such as 3-phosphoglyceric acid, phosphoenol pyruvic acid, or pyruvic acid that is generated during glycolysis, or 2-oxaloacetate acid or oxalic acid that is during TCA cycle acts as the amino acceptor. Aminotransferase enzymes facilitate this reaction. The majority of transamination reactions are reversible following the interchange of amino group between amino donor and amino acceptor compounds. However, exceptions exist in specific cases such as the reactions involving glutamic acid and glyoxylic acid, as well as serine and glyoxylic acid, where the reversibility is restricted. In these instances, the direction of the reaction is favored toward the formation of a specific product, influencing the overall balance of transamination process. As you can see here that aspartic acid and oxaloacetate is converted into glutamic acid and oxaloacetate in the presence of enzyme glutamic aspartic aminotransferase. Aminotransferases depend on the pyridoxal phosphate, that is vitamin B6, as a crucial cofactor for their enzymatic activity. This cofactor PLP, that is pyridoxal phosphate, forms a covalent bond with the protein, that is EPLP, at the enzyme's active site through a Schiff base linkage with the E amino group of a lysine residue. PLP undergoes reversible transformation during the catalytic cycle. In the process, the incoming amino acids donates its amino group to the aldehyde form of pyridoxal phosphate. Subsequently, the amino acids is converted to the corresponding keto acids and it exits the enzymes enzyme active site. Meanwhile, pyridoxal phosphate, upon receiving the amino group, transforms into its amino form known as a pyridoxine phosphate, as you can see here. The second substance, that is an alpha keto acid, then binds to the enzyme's active sites. In a characteristic ping pong reaction, it receives the amino group from PMP, that is pyridoxamine phosphate, leading to the regeneration of pyridoxal phosphate and the formation of the respective amino acids. This aminotransferase enzyme is present in various cellular compartments, including the cytoplasm, chloroplast, mitochondria, glyoxysome, as well as peroxisome. The distribution of aminotransferase across different cellular compartments highlight its importance in diverse metabolic processes contributing to amino acid synthesis and metabolism in various cellular organelles. All amino acids are originate from intermediate with glycolysis, the citric acid cycle, or the pentose phosphate pathway, as you can see in the figure. Nitrogen is incorporated into this pathway through the involvement of glutamate and glutamine. The pathways leading to amino acid synthesis vary in the complexity. While 10 amino acids are closely related, being just a step or a few steps away from a shared metabolite, the biosynthetic routes for others, particularly aromatic amino acids, are more intricate. A useful way to organize these biosynthetic pathways is to group them into 6 families corresponding to their metabolic precursors or alpha ketoglutarate family, in which the glutamate, glutamine, proline, and arginine is included. The next one is the 3-phosphoglycerate family, where serine, glycine, and cysteine is included. And then we have an oxaloacetate family, where aspartate, aspargine, methionine, threonine, and lysine is included. Besides that, there is a pyruvate family, which has a alanine, valine, leucine, and isoleucine. Then we have a phosphoenol pyruvate and erythrose 4-phosphate family. Here we have a tryptophan, phenylalanine, and tyrosine. And the last group family is ribose 5-phosphate, in which the amino acid histidine has been added. So now we will discuss the biosynthesis of amino acids of all 6 families. The glutamine, proline, and arginine are basically synthesized from alpha ketoglutarate, as we have discussed earlier in ammonia assimilation. Glutamine synthetase combines ammonium with glutamate to form glutamine. Proline, a cyclized derivative of glutamate, is formed through a series of enzymatic reactions. The initial steps involve the reaction of ATP with gamma carboxyl group of glutamate, resulting in the formation of an acyl phosphatase. NADPH or NADH then reduces the acyl phosphate to produce glutamate gamma semialdehyde, as you can see in the figure. The gamma semialdehyde undergoes rapid spontaneous cyclization. Subsequently, the cyclized intermediate is further reduced, ultimately yielding the final product, that is proline. Arginine, another amino acid, is synthesized from glutamate through ornithine and the urea cycle in animals. Ornithine could theoretically be produced from glutamate gamma semialdehyde via transamination, but the spontaneous cyclization of the semialdehyde in the proline pathway hinders a sufficient supply of this intermediate to ornithine synthesis. Bacteria have de novo biosynthetic pathway for ornithine and arginine, mirroring some steps of proline pathway, but incorporating two additional steps. In the first step of this bacterial pathway, the alpha amino group of glutamate is blocked through acetylation, requiring acetyl-CoA. After the transamination step, the acetyl group is removed, resulting in the production of ornithine. So, as illustrated in the figure, the ornithine undergoes conversion to glutamate gamma semialdehyde through the action of the ornithine alpha aminotransferase enzyme. The resulting semialdehyde is spontaneously cyclized to form 1-pyrroline 5-carboxylate. Subsequently, 1-pyrroline 5-carboxylate is further transformed into the proline, completing the proline biosynthetic pathway. Unlike some organisms, mammals lack the pathway which has been illustrated in this figure for arginine synthesis. In situations where dietary arginine or protein turnover fails to provide sufficient arginine for protein synthesis, the ornithine alpha aminotransferase reaction operates in the reverse direction, leading to ornithine formation. Ornithine is then processed into citrulline and arginine through the urea cycle to fulfill the demand for arginine in mammals. So, moving ahead to the next family, that is a 3-phosphoglycerate, gives rise to a serine, glycine and cysteine. Now we will discuss their biosynthesis in detail. As you can see in the figure, central pathway for the serine biosynthesis remain consistent across various organisms. This process initiates with the oxidation of the hydroxyl group in 3-phosphoglycerate through the action of a dehydrogenase utilizing NAD to produce 3-phosphohydroxypyruvate. Following this, transamination occurs with glutamate, leading to the formation of 3-phosphoserine. Subsequently, phosphoserine phosphatase catalyzes the hydrolysis of 3-phosphoserine, yielding free serine. It's important to note that serine consisting of 3 carbon atoms acts as the precursor for glycine with 2 carbon atoms, achieved through the removal of a carbon atom mediated by serine hydroxymethyl transferase as already mentioned in the figure. This intricate pathway highlights the interconnectedness of amino acid metabolism and its vital role in cellular processes. Moreover, methylene tetrahydrofolate is generated through the acceptance of the carbon 3 from serine by tetrahydrofolate, leading to the formation of methylene bridge between N5 and N10. This process results in the production of N5 and 10 methylene tetrahydrofolate. Notably, this overall reaction is reversible and involves the participation of pyridoxal phosphate as a cofactor. In vertebrate liver cells, an alternative pathway exists for glycine biosynthesis. This pathway involves the reverse of the reaction and is catalyzed by glycine synthase, also known as glycine cleavage enzyme. In plants and bacteria, the synthesis of cysteine, the methionine as discussed later, involves the production of reduced sulfur from environmental sulfates. The pathway depicted in figure initiates with the activation of sulfate in two-step process, ultimately yielding 3-phosphoadenosine 5-phosphosulfate. In short, it is called as PAPS. Subsequently, this PAPS undergoes an 8-electron reduction, resulting in the formation of sulfide. This sulfide is then utilized in a two-step pathway for the synthesis of cysteine from serine. Then we have oxaloacetate and pyruvate, which synthesizes about 9 amino acids such as aspartate, asparagine, methionine, lysine, threonine, alanine, valine, leucine and isoleucine. Alanine and aspartate are formed through transamination from glutamate using pyruvate and oxaloacetate as precursors respectively. Asparagine synthesis involves the amidation of aspartate with NH4 provided by glutamine. These amino acids are considered non-essential and their uncomplicated biosynthetic pathways are present in all organisms. So let's stop here the today's lecture, and in continuation with this, the synthesis of other amino acids will be discussed in the coming lecture. Thank you.